So good morning, everybody, and welcome to Grand Rounds. We're going to have two more Grand Rounds before we pause and recess for August, and then resuming again in September. Next week, there'll be the annual June Jackson Christmas Grand Rounds, which is something that Patrice Malone has initiated in the context with Melissa Arbuckle and Jean-Marie Bradford as part of the June Jackson Christmas Summer Diversity Fellowship program, where we we offer opportunity for medical students to come to train, get some experience in psychiatric research. And the speakers will be Ayana Jordan from Yale, Utibe Essien from University of Pittsburgh, and Iraka Bath from UCLA. It's a good thing we're doing it virtually. And then on July 28th, the last of the year, I guess somewhat appropriately, we have Shanna Sachs, who is the co-director of the MGH Center for Gun Violence Prevention at MGH. So I guess given what's going on in the country in terms of the surge in gun violence, that's an appropriate topic to end the academic year on. Also, just to mention, and I can't reveal the outcome because the results are embargo, but hospitals make a big deal about the US News and World Report rankings that come out every year. They just have been released, at least released to the institutions with an embargo to the public, so I can't tell you what they are, but they'll be coming in July 27th. So it's something to look forward to, not that it's be all and end all, but it's a significant kind of shout out that occurs every year. Also, I just want to mention that, well, let me go into sort of what is an important thing to mention before the last thing. So COVID, that's the topic of the year, verging on a year and a half. So the COVID pandemic seems to have settled into kind of an undulating pattern. We had the surge beginning in March 2020, subsided over the summer, then over the fall, it increased again and then peaked during the winter. And then as the vaccines were rolled out and as the seasons changed, it declined. You'll recall that at our peak in April 2020, the number of people in the NYP hospital system with COVID was 2,000. Currently it's 65. So you can see how far it's declined, but it's doing so very, very gradually. Just the numbers drop by single digits every couple of days, and then they bounce up by a couple of digits every couple of days. So it's not a linear or a decelerating curve. And what's happened is that the Delta variant has proved to be very contagious, not necessarily more lethal, but very contagious. So what we have now is a pandemic among the unvaccinated people. Unfortunately, there's a large proportion of them. So all of the deaths and almost the vast majority of the infections are occurring in unvaccinated people. There's some, but very few breakthroughs. There are also the developments of certain adverse reactions that are occurring, albeit very rarely, but significantly. So myocarditis in young people, but not severe myocarditis that results in persisting myocardopathy. And also some very rare cases of Guillain-Barré. The problem is that with a significant portion of the population unvaccinated and exposed and contracting infection, they provide a vector for mutation and the prospect of new variants. And the new variants so far, the variants so far have been contained by the vaccine. However, there's always the possibility that new variants won't be. And in that context, there's been discussion, and it's been kind of a confusing discussion between the CDC, Dr. Fauci, the pharmaceutical companies, particularly Pfizer, about whether a booster might be necessary. The latest that Dr. Fauci said was that it's not now, but it very well may be later. And the other thing that's being considered, but again, is not decided and not recommended, is the possibility of combining vaccines of different mechanisms. So the mRNA vaccine with the adenovirus vector vaccine from J&J, because they act in different ways, and it could be relevant in providing a broader spectrum of immunity for the variants that may attach to other things than the spike protein on the virus. So all of this serves to prolong the process of normalization. I mean, we're doing very well locally, and the city is doing okay, although not great in terms of the infection rate continuing to decline to zero. But we need to keep doing it. And we have the guidelines still in place with regard to mask, distancing, telecommuting. And we need to continue them as we try and increase our on-site presence and normalization of activities, but it's not something that clearly there's an endpoint in sight, and we know we're going to sort of progress to that. The last thing I'll say is that if it was up to me, we'd have mandatory vaccinations. The hospital has mandated them, the university has mandated them, the state has not. And our vaccination rate is good, but not 100%. And I feel everybody should be vaccinated. So if you haven't, you better have a good reason, meaning some medical explanation or religious, justification. But if we're vaccinated, then we're safe among ourselves. And if patients are coming in for research or for treatment, they'll want to know, are your staff vaccinated? So if you have any questions about that, Alicia, our infection control nurse, can answer them, Rebecca Owens or Evelyn Attia. Lastly, I just want to say, we're introducing our Grand Round speaker, that you probably have noticed that Grand Rounds in recent years has worked very, very smoothly, efficiently, with a real high quality of speakers and programs, and it's largely because of Jonathan Posner. Jonathan's been the coordinator of the program and has done all the heavy lifting and organizing and orchestrating it, and I just get to come in and introduce and pontificate. But unfortunately, not unfortunately, but unfortunately for us, Jonathan's going to be leaving at the end of the summer and taking a new position at Duke as vice chair for research with our own Maura Wren, who's the chair there. So we will be trying to fill Jonathan's considerably large shoes in some similar capacity going forward. But I just wanted to publicly acknowledge that and thank Jonathan for all the work he's done in this regard. So our speaker today, again, and what prompts my recognition of Jonathan in this way today, as opposed to next week or the week after, is because his area of expertise was the source of the invitation to Tomas Paus, who's our speaker today. So Tomas hails from the Czech Republic, but he did his where he received his MD there, and then he came to he received his MD and his PhD in physiology in Prague, and then came to Canada and to Montreal at the Montreal Neurological Institute of McGill University, a legendary bastion of neuroscience research, and did a postdoctoral fellowship in cognitive neuroscience. And we've seen over the years new disruptive disciplines emerge. Molecular biology, molecular genetics certainly has been cognitive, computational neuroscience, things that have really been synergistic in bringing together distinct disciplines. And Tomas seems to have had an inclination towards that approach very productively. He initially worked on functional and structural organization of the cerebral cortex using a variety of approaches of studying patients with brain lesions and with functional and structural neuroimaging and brain stimulation. And then he turned to a more integrative approach with studying not just individual subjects, but populations of subjects, integrating the disciplines of epidemiology, neuroscience, and genetics in a new discipline of population neuroscience, so to speak. And most recently, he's continued this and contributed to the field of neuroscience in two ways. One, methodologically, by introducing a number of innovations for the study of the human brain, including voxel-wise analysis of structural brain images and combining brain stimulation and imaging. And then, in addition, let's see, I'm getting ahead of. In addition, by trying to understand the function of specific brain regions, and most specifically the anterior cingulate cortex and the processes underlying changes in white matter during the maturation of humans through adolescence into young adulthood. I should have asked him before, but maybe he'll comment in the course of his talk of whether in this work he's been involved with Alan Evans and the atlas that the Evans Laboratory has constructed, which has been very helpful. But it's a pleasure to have him here today to share his work and to talk to us on population neuroscience of the developing brain. So please virtually welcome Tomas Paus.
Thank you very much. Thank you for the invitation. Nice introduction. Really glad to meet you, Dr. Lieberman. Thank you, Jonathan, for extending the invitation, and to Dr. Waxman being interested in our work. And I'm impressed by the number of people who are logging in. So if you stay with me for the next 45 minutes, hopefully you'll be able to learn a little bit from the kind of work that we do. Now, as I was saying, I wish I was in New York. I miss the city, and hopefully this pandemic will be over soon, so that we can cross the border again. So today, what I will try to do very briefly at the beginning, I will explain how I'm thinking about population neuroscience. Then I'll spend most of my lecture on gray matter, and I'll tell you in more detail about two most recent studies that we have completed that are relevant, as I will describe, to psychiatric disorders in collaboration with Enigma Consortium. But I will finish with my really sort of love and interest in white matter, but that will be a very, very small part of the lecture. So let's start with the population neuroscience. Well, to this audience, I don't need to talk about the motivation for this work. It's really because the disorders that you work on really contribute to many years lost to disability. And there are three main reasons for it. Those disorders are prevalent, very often they are chronic, and the third one is they start early. They start 50% before the age of 14, 75% before the age of 24, 25, roughly speaking. So most of the psychiatric disorders, I hope you would agree, can be thought of as disorders of brain development. And that's really what I'm interested in, to understand what shapes human brain development. And of course, there are only two possibilities. It's our genes and our environment, and of course, the interplay between the two. And as you very well know, I mean, the complexity is staggering. The brain is complex, the number of genes is high, and the amount of potential influence is coming from our environment from before we are conceived onwards, it's staggering. And so one of the approaches that we have started applying over the past now almost 20 years is what I call population neuroscience, to try to get at the complexity by not zooming in a univariate way on one particular aspect, but try to take the complexity in stride and do that in the context of, in my work mostly, prenatal or perinatal development, and also during adolescence. And that's where population neuroscience comes in, as really a combination of three main disciplines, if you like. Two that are about exposures, talking using the terminology of epidemiologists, where epidemiology covers mostly, if you like, the external exposures, and genetics, you can think of it as covering the internal exposures. And then the outcomes, for me, it's the brain. For you, it's, of course, behaviors just a small distance away from it. Now, let me go briefly over the three. So first, historically, and it's quite, you know, 2 years ago, people did not care much about pandemics, but certainly epidemiology begins with epidemic of cholera in the mid-1800s, where London physician John Snow noticed geospatial relationship between the source of water and death of cholera. So those who got water from this pump died. Those who got water from this pump lived. Now, that was 20 years before we knew what the cause is, Vibrio cholera, that was discovered 20 years after this in Germany. So it's really the first example of a pure correlation, geospatial correlation between a potential agent or a behavior in this case, where you got your water, and disease, with no causality, an observation, but very important one. And epidemiology then moved into different areas, in particular cardiovascular health, but also into our area of psychiatric disorders. And I think that this one is a nice study based on health records in Sweden that show that there is a fairly strong relationship between preterm birth and psychosis. As you can see, if you were born before 32 weeks of gestation, even in the crude and corrected model, you were much more likely to be diagnosed with bipolar affective disorder by the age of 23, and the N is over 1 million young Swedes. But again, it's just an observation. We don't know anything about the directionality of that relationship. We don't know anything about causality, but it's a good starting point to get into. And I think epidemiology is an amazing source of knowledge about phenomena, important, especially from the public health point of view. Now, genetics, of course, there are many different ways one can think about it in terms of genetic variations. I won't be talking much about those. In terms of regulation of gene expression with, for example, DNA methylation epigenetics. Again, I won't be touching on it today, but I will spend some time on transcriptomics, gene expression, and gene expression as a tool to understand the phenotypes that we measure with MRI. And that's where I will spend a big part of this lecture. And finally, of course, neuroscience, or in my case, mostly neuroimaging, as a way to really observe what's happening in the human brain. And of course, there are many different tools, traditionally both on the functional side, but also the structural side. And today, I mean, overall in my work, I put my money on brain structure for many reasons, including the fact that structural-based phenotypes are highly reliable, the way that we can measure them, that they allow us to assess in an even, unbiased way the entire brain, and also they are relatively straightforward to acquire in large numbers. And today I will really talk mostly about the simplest possible type of structural image, T1 weighted image, that provides us with a very good contrast of gray and white matter. Now, I put here days or weeks or years, and that's important because at least to some extent, brain structure, I will show you, of course, we are born with certain brain structure, but also brain structure reflects our experience, and we should not forget about that. So let me move now into describing to you two particular studies or paradigms that I hope will be helpful for you to understand why looking at the brain anatomy is actually useful for a psychiatrist. So let's start with a few numbers. You all know that obviously cerebral cortex is the biggest part of our brain. In fact, it's about 80% of brain mass, the gray and white matter underlying cerebral cortex, of course, large number of neurons and a fairly large number of non-neuronal cells. And the ratio when we combine gray and white matter of neuron to non-neuron is about 1:4, almost 1:4. And I'll come to, in fact, the cellular composition of the cerebral cortex very shortly. Now, when we look at the cerebral cortex, certainly on an MRI image of the human brain, we can see that in two key dimensions. One is, of course, the vertical one, and that's the cortical thickness. But the other one is the tangential growth of the cerebral cortex, and that's the surface area. And in the next 20 minutes, I'll be talking about both thickness and surface area. I will start though with the thickness. Now, in the first study I'll tell you about is a study where we were really interested in psychiatric disorders, cortical thickness in psychiatric disorders, but in particular in the neurobiological underpinnings of what we might see in those disorders. And that's where I'm introducing the term virtual histology, and I'll tell you all about it. But basically, the bottom line, of course, we would like to do proper histology of those brains, but we can't. We certainly can't do that in living individuals and in thousands of individuals. Because, of course, MRI gives us some idea of what's different at a gross morphological level, but that's not good enough. We need to know, of course, what underlies those MRI-derived phenotypes. And that's what I will try to tell you about in the next 20, 30 minutes. So the first thing that we did in this case, we entered into a collaborative work with participants in the Enigma consortium that contains many of you are members or have worked with the consortium in different contexts. And the consortium has a number of working groups, including working groups on different psychiatric disorders. And in this particular case, what we did, we approached leaders of those 6 different working groups of the 6 psychiatric disorders listed at the top. And then we approached them and asked them whether they would be interested in assessing regional variation in cortical thickness as a function of the disease status. So we sent out the scripts to about 140 different cohorts covering those different disorders and asked them to run a model that is identified. Here we received the results, the statistical summaries, and then ran a meta-analysis to identify regional differences in cortical thickness between cases and controls for those different disorders. And the main goal, though, of the study was not just to see the profiles, but to use virtual histology to understand what might be at a cellular level underlying those differences. Now, let's see first what we got. You see the number of cases and controls you see on the right. So we had a very nice sample sizes for all those disorders. And here you see the group differences in cortical thickness for each of the 6 disorders as a function of 34 cortical regions. You know that some of you may know that with free surfer, using a particular segmentation, you get values of, for example, cortical thickness or surface area for each of the 34 regions per hemisphere. So they are grouped by lobe here and just lined up along the x-axis. So the obvious thing that you see right here is that patients with schizophrenia showed much lower thickness than the controls in their cortical thickness, less so for the other conditions, bipolar here, OCD here, etc. Okay, so now this will be our input into virtual histology. You also see right away here that there might be some similarities in those interregional profiles of group differences across the different disorders, and I'll come back to that in a minute. Now, virtual histology. Let me take a short detour to explain what we are doing here, because that's really important to understand. Obviously, human cerebral cortex consists of a number of cell types. The most common neuronal cell type are pyramidal cells. 70% of all neurons are pyramidal cells. About 30% are interneurons. When it comes to glia, non-neuronal cells, the majority are oligodendrocytes, a fairly large number of astrocytes, microglia, but also the other glial cells, ependymal, endothelial, and neural cells, so the vasculature here. Now, how do we go about actually trying to get a feel for that cellular composition without cutting up those brains? And that's where virtual histology comes in. The idea is very simple. We will use cell-specific gene markers to look at profiles of gene expression across the same 34 regions using gene expression data from the Allen Human Brain Atlas. Now, the first thing that we need to do, though, is to check whether we can actually use data based on 6 hemispheres only to relate them to, of course, our MRI scans obtained in a different population. So we need to know whether those profiles are consistent. And to check that, we introduce basically a 2-stage filter. We are working only with the genes that have profiles across the 34 regions, very similar to the median profile. The black is median profile, the different dashed lines are the 6 donors. And this is an example of a consistent profile. You can see that everyone is more or less following the median profile, okay? And we then went to the second step where we also checked whether those profiles are very similar with another database, the BrainSpan. And at the end, we end up with only 2,500 genes that we consider to be consistent and reasonably representative, therefore, of the profiles that we would find in your brain, my brain, in any other brain, right? So they are now, but remember, the consistency refers to a profile. It's not about the absolute values of gene expression in a given region of the cerebral cortex. It's the profile of it. In the same way as we can look at neuropsychological profile of symptom of psychiatric disorders, it's about the profile, not the absolute value of one of the members of the profile.
Tomas, just so that I can clarify for myself and anybody else who's not clear. So you're taking postmortem tissue from 6 people or so, and you're looking at the gene expression, and from that, you're then deriving or sort of inferring that the specific anatomic regions in the imaging data that you collect will be reflecting the activity of those genes or the gene expression in those anatomic regions. Is that correct?
Well, more or less. Let me come back to that. But it's really, you need to think about the gene expression in this case is more of a constitutive gene expression that is essentially marking the phenotype of a particular cell type, a phenotype of an astrocyte, or kind of maintaining the cell as an astrocyte versus microglia versus pyramidal cells.
So you're using that as an index of the cytoarchitecture of the region?
Essentially. And let me, there are two more slides that will clarify that a little bit more, okay? Because that's the next step. We need to know what the cell markers are. And there we went to a different data set with single cell RNA that actually identified a set of genes that are specific to each of those cell types. Okay, so that's where we gained the cell specificity. Now, the number of genes in brackets are now the numbers that are consistent based on the definition that I explained to you. So now we have 103, for example, genes that are specific to this type of pyramidal cell. And I would think of it now as genes that are maintaining the cellular identity of that cell from birth to death. That is not perturbed, for example, by age, by psychiatric illness, by sex, right? That's the assumption. So that's where the consistency comes in. And so, as you said, it's basically a genetic fingerprint of cytoarchitecture in that sense, right? And what we do next, we take those cells, those gene expressions. So now we have the distribution across the 34 regions, distribution of cortical thickness or group differences in cortical thickness. In the same 34 regions, we have the values of the expression of each of those marker genes. I give example of one particular gene, marker of astrocyte. And then across the 34 regions, we correlate the two. So each dot is obviously not a person because we do not have, of course, gene expression from the people in whom we have MRI. Each dot is a brain region. There are 34 of those dots, right? Because there are 34 regions. And it shows that in this particular case, the expression level of this particular gene correlates strongly with the thickness across the 34 regions. And we repeat it over and over for all those panels of gene expression, and then test statistically whether one particular set of genes, one particular panel, shows a stronger relationship with our phenotype of interest across the 34 regions.
Tomas, just one other question. So would you assume in your cortical thickness measure on the y-axis that if you were looking at one of the genes expressed from oligodendrocytes, that it would be uncorrelated and very little expressed?
That's an empirical question, and we have published a few papers on it. And in this particular case, yes, that's true. In fact, oligodendrocytes do not contribute to cortical thickness. They might contribute to cortical thinning, but that's it. Yeah, so that we've published a few papers on that, in particular during adolescence, but also across the lifespan. Okay, so this is very important principle to understand. We are not correlating across individuals, of course, because we don't have their gene expression in the brain. We are correlating across regions. That's how we can bring together those two sets of data, MRI-derived data and gene expression data. Okay. Now, when we do that in our 6 psychiatric disorders, and we do that for the group differences between cases and controls, you can see that pretty much in all psychiatric disorders, this type of pyramidal cells that is typical for large dendritic arbor, in all those cases, across regions, it relates to the extent of those group differences. And the directionality tells us that regions with more of those cell types, more expression of those cell-specific genes, are the regions that show larger group differences between patients and controls. Okay? But to some extent, it's also true about astrocytes, and about microglia, with few other exceptions here and there. Okay, but let's focus now on those pyramidal cells. We can ask also what's going on in terms of the commonality of the profile. Remember, I pointed out to you that there are some similarities in those interregional profiles of group differences across those 6 disorders. So we use principal component analysis now to capture that shared common profile. And as you can see here, that principal component 1 that is shared across those 6 disorders is telling us that pretty much in all 6 disorders, we see very little difference between cases and controls in the visual cortex. And we see the largest difference, again, in all 6 disorders in the temporal cortex, right? So now we can again run our virtual histology on this PC1, and not surprisingly, of course, we get pretty much what we got before. We get again those pyramidal cells. And so in the next step, we ask what other genes are co-expressed with these pyramidal cell genes. And for that, we go to 5 different databases of gene expression over 500 brains. And when we do this co-expression with those pyramidal cell genes, we get roughly 400 genes that we decided we plot now their expression as a function of age, from prenatal period to postnatal period. That's from the Brainspan Atlas. And as you can see, there are essentially 2 clusters. Genes that are expressed prenatally, but then go down in terms of their gene expression, and genes that show the opposite, low before birth, higher after birth. And then we can use gene ontology on those 2 clusters and ask what kind of genes are really dominating the 2 clusters. And not surprisingly, for those prenatal genes, we see a lot of terms that have to do with the initial development, whether it's axon development, axonogenesis, dendrite development, etc. On the other hand, for the postnatal genes, we see a lot of gene ontology terms that have to do with neurotransmission and synaptic plasticity. Okay, so functioning of the brain after birth. So to sum it up, the way that I'm thinking about these findings is that it's like piano keys and piano player, a pianist here. The pyramidal cells are the piano keys. These are the cells and genes that are, of course, very close to the phenotype of interest, cortical thickness. They are the ones that are highly arborized. They are running all those computations. But they are not the cause, of course, here. They are not the ones who are being shaped. It's the other genes or maybe environment that who are the pianists, who are playing on those piano keys and producing music, or producing, of course, brain function. So that's the way that I like to think about the results of that first study. Okay. Now let's move to the second one, and that's about surface area and what I'm saying here, looking back in time. And for that, we need to talk about what we are talking about is really virtual ontogeny. Again, we have our 34 regions. We will measure surface area in a subset of those regions, as I will explain in a minute. And then we will try to figure out which genes, which cell types early on in fetal development predict group differences in surface area in different psychiatric disorders. Okay? Now for that, I need to emphasize and point out to you that surface area, that's not surprising to you, the tangential growth of the cerebral cortex happens primarily before birth and during the first 2 years of life. So once I look at your brain, my brain, in terms of the surface area, it's pretty much where it was when I was 2, 3 years old. It's frozen in time. And that's very important, because then I can take this phenotype and ask what determined it in terms of those early developmental processes. Okay.
Tomas, is surface area and cortical thickness highly correlated?
Not at all. They don't correlate with each other. They do not. And also when you look, for example, during adolescence, surface area is flat, cortical thickness goes down. These are two uncorrelated phenotypes. And just very briefly, you all know about the radial unit hypothesis of Paus-Rakic. The tangential growth of the cortex is really about radial glia, about the neuroepithelial cells and the progenitor cells, intermediate progenital cells. The more activity you would have, more symmetric and asymmetric divisions, the more ontogenetic columns you would have, and the more surface area you would have, because you are giving birth to all those neurons that are then migrating to the cortex. So these cells, the outer radial glia and intermediate progenitor cells, are really the key for the tangential growth of the cerebral cortex. And you can see that that's, of course, happening towards the end of the first trimester and the beginning of the second trimester.
Not to throw you off your talk, but does the prospect or controversy about neurogenesis, relevant to this, this process or this consideration?
Well, it depends who you talk to. Paus-Rakic will tell you no.
Right.
Right?
He'll do more than that. He'll kill your career.
Yeah. So I think that, and I'm more on his side in that neurogenesis, yes, it's a reality, but it's a reality in the olfactory system and potentially hippocampus. It's not a reality in the cerebral cortex. And certainly not to the extent that it would ever influence the surface area, in fact, after birth. I don't believe that would play a role. Okay, so we are really restricted, I think, in terms of the major change in surface area to this period of prenatal development. But good question. So now we use again the same approach. We will be again correlating profiles of group differences now in surface area with profiles of gene expression, but now it will not be gene expression of the cell-specific types that we identify for virtual histology. We need to identify genetic markers of those cells that are happening, that are relevant for brain development, for cortical development. For reasons that have to do with availability of gene expression values during fetal development, we are limited to 11 cortical regions rather than all 34. So now let me describe to you how we develop this virtual ontogeny pipeline. We start again with single cell RNA, but now it's a single cell RNA from 5 individuals, gestational age between 6 and 22 weeks. So it's a fetal cortical tissue, over 189 cells that were analyzed in terms of single cell RNA and then clustered to identify essentially markers of those cell types. You see, for example, these are genes that are specific to intermediate progenitor cells, genes that are specific to radial glia, excitatory neurons, etc. So now we have our gene, we have our cell-specific markers that are relevant for this developmental period. Now we need, of course, the data on regional variation of those markers. And there we go to the brain span or the SIGIN code, where we have those 11 regions that have been sampled at different periods of time, including between 10 and 22 post-conception weeks. And finally, we go to Enigma and extract surface area and group differences in surface area for exactly the same 11 cortical regions. And then, of course, what we will ask is what is the spatial relationship between the expression profiles based on this data set and profiles of group differences in surface area in this data set. Okay, again, the correlation of profiles across, in this case, 11 regions, cortical regions. And we go back to Enigma Consortium, now leaving out OCD, but the other disorders are here. And we have added also the ABCD study that we dichotomized into those with top and bottom 20% of total CBCL score. Okay, so we have a community-based sample, not, of course, clinical diagnosis, but extremes in terms of psychopathology. And we again run the scripts that tell us about group differences based on a disease status in cortical area. We get our summary statistics, we do our meta-analysis, and then apply virtual ontogeny. And here are the results of those meta-analyses. Now I'm plotting it in a way that if you are above 0, the controls have higher surface area than cases in a particular region. Okay, as we would predict, in most of those disorders, controls have higher surface area than patients with those conditions, including, in fact, the difference between the top and bottom psychopathology for the ABCD study. You can see also that we see very little, fairly flat profiles for bipolar disorders and major depression when it comes to surface area. Remember, that's very different for cortical thickness. And for autism spectrum disorder, they hover around 0, but they do have a profile. There is some ondulation on this, and I'll come back to that. So now let's have, but you see also, and I have ordered it in a particular way, that there are certain regions that show much less of a difference than other regions. And so we ran a cluster analysis on this, on those group differences. And in fact, the best solution were 2 clusters that group together regions that are primarily multimodal, and then in the second cluster, regions that are primarily unimodal. And when we compare the group differences obtained for cluster 1 versus cluster 2, we see that certainly for ADHD, for schizophrenia, and for the ABCD data set, the group differences are much larger in what I call here multimodal regions as compared with the unimodal regions. So that's already interesting that when it comes to group differences in surface area in several psychiatric disorders, the differences are larger in associative cortex or multimodal cortex than, for example, primary motor, primary somatosensory, primary auditory, or visual cortices. Okay. Now, let's have a look at what's happening when we divide the brain, the cortex in this way, what's happening with gene expression of those key cell types during early development. And this is really important. You see that for those proliferative cells, radial glia and intermediate progenitor cells, the gene expression is higher for the cluster 1, but it's lower in differentiated cells, whether it's excitatory interneurons or, for example, endothelial and neural cells. And keep that in mind. So something is very different in the developmental trajectory for the proliferative cells versus the differentiated cells during that early period of development. So what did we find when we now related in space the group differences in surface area for the different disorders with the expression profiles of those different cell types during this early before birth period, right? And that's where I'm showing you the strength of the correlations. You can see that the overwhelming effects are found for those proliferative cells. And the correlation, that these are the interregional correlations between their gene expression and group differences, are telling us that regions with more expression of those proliferative cells are showing larger differences between cases and controls, and the opposite for the differentiated cells, excitatory, and to some extent also endothelial and neural cells. Okay? So clearly, what's going on differentially across different cortical regions during that very early period of development is, in particular, in terms of the proliferation, is predicting now group differences in individuals who eventually become diagnosed with a psychiatric disorder.
So, Tomas, sorry to keep interrupting, but to clarify, the expression, the gene expression that you're looking to correlate with surface area in individuals that express a disease phenotype. If you just look at schizophrenia, which in terms of the anatomical measures that are derived from structural imaging, that's an illness that has among the largest differences, but it has the smallest correlations in terms of the gene expression. Am I understanding that correctly?
Schizophrenia is here, right?
It's
and
green.
it shows that the relationship.
I'm sorry. I was looking at the wrong color.
Yeah, no, it's in fact schizophrenia and ADHD have the strongest relationship. To some extent also ASD, even though the profile was close to zero, but it was, I pointed out to you that they were interregional variations in that group difference, right? Now, I can tell you that even though the CBCL, the ABCD has low effect size, it's highly significant, which suggests that it's highly consistent, even though the relationship is relatively weak in terms of the size of that relationship.
Excuse me, did I tell you I was colorblind?
I should recode it. Yes. Okay. So yes, schizophrenia is the number 2 here.
Thank you.
Okay. Now the last point in this story, what we wondered, could we get at the issue of environmental effects on brain development and how it intersects what we are seeing here, how it intersects with the role of those proliferative and differentiated cells, gene expression. And so we went to the literature, and there is a nice paper in Lancet Psychiatry that calculated odds ratios when it comes to psychosis of the relationship between different risk factors, such as birth weight, hypoxia, etc., and the risk of psychosis. Okay, so this is the Z-score. These are the risk ratios. Okay, so now we know, as you know, that yes, those different factors that are the prenatal factors may influence the outcome, long-term outcome. And we wanted to know whether at all we can bring this together with what I just told you, the story of the proliferative and differentiated cells. How did we do that? Well, for each of those factors, we use either gene ontology or existing GWAS data to identify a set of genes that have something to do with that factor. So for example, genes that that have to do with congenital anomalies, 558 of those genes, or genes that are related to hypoxia, 359 genes, response to hypoxia, etc. And then we simply tested for enrichment between of those genes in our cell-specific developmental genes. And I found this incredibly interesting because it tells us that these conditions seem to be intersecting with the proliferative genes, especially radial glia, whereas these conditions, blood pressure, preterm birth, are intersecting with the vascularization genes, right? And also the preterm birth and eclampsia is showing the same profile. So that tells me that there are two different pathways that may lead to variations in surface area and eventually have something to do with the risk of mental illness. One pathway that cuts across the proliferation, another pathway that cuts through vascularization, of course, during the pre-development before birth.
What are you considering as congenital anomalies?
They were any kind of congenital anomalies that basically, this is not that we are identifying them, it's the gene ontology analysis that curated genes as associated or not with any kind of congenital anomaly.
I see.
I cannot give you a list of them. I would have to go and look at the curation.
Yeah, okay. You mean like these DiGeorge syndrome or things of that sort?
Yeah, anything. So let's go now back and summarize. So clearly, proliferation during early period of brain development before birth is one of the common mechanisms that may be affected and abnormal in some way in individuals who eventually develop psychiatric disorders. And that process seemed to be particularly important for cortical regions that have multimodal character and function. Now, if we go back to our piano and piano keys analogy, now the piano keys are our proliferative cells, our genes that are specific to proliferation. The piano players, the pianists, are, for example, the environmental risk factors that I just described to you, and or, of course, other genes that interact with the genes that are close to the processes underlying the tangential growth of the cerebral cortex. Now, I can stop here if you like, because I think that we are kind of running out of time, or I can spend another 5 to 10 minutes to, well, 5 minutes to talk about white matter, but that's up to you.
Jonathan, what do you want to do?
I think the extra 5 minutes to hear about the white matter would be terrific. So please do. Okay,
so very briefly, so we are shifting gears here. We are talking about those thousands of kilometers of axons in the human brain. And it takes me back to the initial studies from our first cohort that started almost 20 years ago, the Sagana-U study, where we showed that very clearly the relative volume of white matter increases sharply during adolescence between the age of 12 and 18 in males, and less so in females. And it's not simply a delayed trend, it's really sex-specific phenomenon. So what we wanted to do is to check whether testosterone has anything to do with it. And we used functional polymorphism in androgen receptor gene, the number of CAG triplets, to test the possibility that yes, testosterone in males is influencing the volume of white matter during puberty, a good assumption to make. So we are injecting a little bit of causality into the study of that relationship. And as you can see here, boys with the less efficient androgen receptor gene and therefore androgen receptor show less of a relationship between the volume and testosterone levels as compared with boys with the short, more efficient version of the androgen receptor gene. But of course, the interesting question is, what is it that is increasing? Is it myelin? Everyone would always say, oh yes, of course, myelination is going on during adolescence, so the volume increase must be reflecting myelination. Well, not so, because we had another sequence, magnetization transfer ratio, which to some extent, at least in white matter, is related to myelination. And if anything, we see the opposite relationship. The MTR is higher in those with low levels of testosterone. So the boys with high levels, we can say, are demyelinating. Of course not, right? So there must be something else going on. And that something else is axon diameter. You can see in the monkey corpus callosum, of course, axons come in different sizes. In fact, this axon, it's not a different magnification, the same magnification as the left side. So there can be axons that are as large as that. Now imagine that in your unit of imaging volume, you have more axons like this. Well, there is less space for myelin, and therefore the MTR values will be lower. Now, so what we are saying is that during male adolescence, what's happening is the axons are bulking up. They are getting thicker. There may be, of course, changes in myelination. We are not saying that there are none, but at least the phenomenon that I showed you, we suggested it's dominated by the radial growth of the axon. And I'll tell you in a minute why we care about this. But first we wanted to verify it, and we did electron microscopy in the rat corpus callosum, measured the axons, and showed that indeed the axon diameter is larger in males compared with females. And we also could do what we could not do, of course, in our participants. We castrated the males just before puberty. And as you can see here, the castrated males have axon diameter that is much lower than that in intact males, suggesting that certainly gonads have something to do with that difference in axon diameter.
Can you androgenize the females?
Haven't tried that, but I would think that that we could, yes. That would be a good experiment to run. Now, why do we care? Well, this is what in the axon, cytoskeleton, neurofilaments, and microtubules. And microtubules are the interesting ones, because microtubules are really the roads on which there are motor proteins carrying different cargoes from the cell body to the synapse, or from the synapse to the cell body. So we have the retrograde transport by dyneins and the anterograde transport by kinesins. Anything that I need down here as a protein needs to be made in the cell body and transported using this system. So you can imagine how important axonal transport is for a proper functioning of our brains. That has been recognized widely by those studying neurodegenerative disorders. I don't think it's as recognized by us studying development and psychiatric disorders. Now, is it the case that axons with larger, well, first of all, does testosterone change axonal transport? And we did a very simple in vitro experiment on sympathetic neurons that we grew in a dish, and then added different dose of synthetic testosterone that cannot be aromatized, so we know that it cannot be converted to estrogen, and we measured axonal transport. And now I'll show you the most exciting movie that you will ever see, and that's that of axonal transport. Now I can see that that vesicle is moving in the axon. I can measure in which direction it goes, I can measure how fast it goes, I can measure how often it stops. So finally, I can see inside the brain here and see at the cellular level what's going on. And that axonal transport was profoundly influenced by adding testosterone. When I measured it 6 hours after the application, the anterograde transport was suppressed. When I waited for 2 days, the transport was accelerated. Now imagine the poor pubertal boy with the testosterone levels going up and down from one day to another. What a havoc it's going to rack onto everything that is being transported back and forth. And that's why I think it's really important to keep that in mind, that this machinery for both the fast non-genomic changes and the slower genomic changes that are induced by different mechanisms may be super important for us being interested in the early vulnerability, for example, to psychosis during adolescence. And so sort of theoretically, what we have proposed is that obviously axonal transport and axonal cytoskeleton are mutually supporting each other. And if you interfere with axonal transport, as I just showed you, by testosterone, but we know stress influences it, cytokines, we know sleep influences axonal transport. If you mess up with it, if you induce fluctuations in axonal transport, you may change, of course, the properties of white matter, but also increase the risk of mental illness. So that's the part that I really want to pursue in the future in terms of the white matter. And to conclude, what I was trying to give you is the sense that population neuroscience is really fun, particularly because you can integrate and go across different levels of analysis. I did not talk about the social level, but we have started some work along those lines, where we go from the organs to cells to molecules and get a better appreciation both of the complexity, but also the potential of where we can go next. And I'll finish up by acknowledging all my collaborators, and particularly the work of my PhD student Yash Patel, who is about to move to NIH for his postdoc, who did the work on gray matter, and my former postdoc Marcia did the work on white matter. Thank you.
Wonderful. Thank you so much, Tomas. That was really just a brilliant fusion of three very, very complicated fields: genetics, neuroscience, and epidemiology, and even with a little molecular biology thrown in there. So really, really inspiring work. I guess, I'll get us started with one question. And I guess in some ways it's sort of a basic or maybe foundational question for population neuroscience. If you think about other areas of medicine, we have population norms. So if a physician wants to look at electrolyte levels in patients, you can immediately reference a normal range and determine whether your patient is outside of that range. I'd love to get your thoughts on how far away we are from having something like that for brain structure. To know whether an individual patient, where they fall in with the norms in terms of cortical surface area, cortical thinning, subcortical volumes. How far are we away from that, and what are the stumbling blocks?
Well, I think that, and we are pursuing it actually, that's work in progress. I think that the concept of profile is going to be very helpful here. Because what we are looking at is not, I think that what we should not, or what is not particularly helpful, is to look at differences in whatever metrics we have for a particular brain region across individuals. I don't think that that's getting us too far. But looking at differences in a profile, a specific profile, like the ones that I showed you, may get us a bit closer, that we are bootstrapping our way into a sort of a major difference between an individual and the population mean. And that's what we are now, in fact, exploring in the context of the surface area, to see whether we can identify, well, obviously statistically, we can identify individuals who differ from the population mean of the profile, more than others. Of course, the prediction is that those individuals will stand out when it comes to psychopathology or certain aspects of genetics. Now, this is still not, it depends how far they stand out. I don't think they will stand out far enough to detect an individual who is at a high risk of a disorder, but at least we should be able to identify a subgroup of individuals who are at a higher risk.
Great. I wanted to turn it over to Dr. Lieberman to see if he has any questions.
Well, let me ask quickly. So just taking a different perspective on this, which it was just amazing work, Tomas. Congratulations. It's really just amazing and very, very inspiring. So structural brain imaging has been a durable methodology that's yielded some informative group differences among different disorders. But as Jonathan was alluding to, it hasn't been able to establish population norms that would allow for any kind of diagnostic or prognostic application. And I remember when I was at UNC and Duke, Alan Johnson did what was called in vivo microscopy, which was an attempt to sort of get to the Jeff Lichtman level of anatomic detail of living animals in this case. You've taken a different approach by looking at the transcriptome phenotype within regions to look at the cytoarchitecture and then track that along stages of development and by disease very ingeniously. So the whole thing is, how do we basically solve the problem of looking into the brain and a living individual in a way that's going to be informative without waiting for them to die or being very invasive? Apart from the surface area, apart from the volume, apart from cortical thickness, and apart from the cellular phenotype, does shape have anything to do with this? I remember a lot of effort went into by computer scientists developing algorithms to measure deformation in shape. Does that provide any useful information in your opinion?
I'm not sure about that. There is a lot of stochasticity, I think, when it comes to shape, even shape in terms of sulci. I have done in the 90s, I did our cyclotron broke down, and so I was stuck with MRI images to do some interesting work, and that's when I focused on morphometrics of cingulate sulcus, paracingulate sulcus, and got an appreciation of shape at that level. And I don't think that, I think that a lot of the subtle differences are really stochastic in nature. And so I would not put too much money on it. What we are doing is we are using other modalities to get at other structural properties of the cerebral cortex. So we published a paper recently with the same PhD student looking at multimodal imaging that we acquired in one birth cohort in Alsbach in England, where we have data on T1 relaxation time, T2 relaxation time, myelin water fraction, MTR, FA, MD in the same set of individuals, 500 of them, and we can derive values of those different structural properties across different cortical regions. And of course, as you would expect, there is again a variation across the cortex. So, but now we have more data to work with, right? We have more values. It goes beyond simply thickness and surface area. It's looking into the composition. It's a little bit like spectroscopy in a way, but right, the same kind of idea, but at a different level. I think spectroscopy would be very interesting if we finally were able to really do it with a small voxel, right? Imagine that you can do spectroscopy in 1 by 1 by 1 millimeter voxel. Then you can, of course, map out chemo architecture, right? And that will be, again, I think, extremely helpful.
Thank you.
Great. So listener wrote in, Jody Weinstein, I noticed in your data on gene expression of the lifespan that a subset of the prenatally expressed genes were also expressed in late teens and 20s. For a disorder like schizophrenia, which tends to present in this age window, would it be reasonable to examine the subset of genes that followed this pattern?
What we wanted to do, yes, I mean, that's the simple answer. I think that what we need is our colleagues who work with those unique data sets of fetal and developmental patterns of gene expression in the human cerebral cortex. We just need to ask them, could you please generate more data so that we can subdivide those developmental periods and repeat this type of analysis in a sliding window. And that would be amazing, right? But it's limited right now by availability of data, because you need data not for one particular region. Of course, everyone, and that's, I guess, Joachim Fuster's and Pat Goldman-Rakic's fault. Everyone is working on prefrontal cortex, but I need more cortical regions. To do what I showed you we are doing, I need a profile. I need the data from at least those 11 regions, if not more. And I need robust estimates for each of those developmental windows. And then I can start asking, okay, so which developmental window is driving most a particular phenotype measured at a particular time in life?
Great. So Ralph Wharton wrote in, wonderful presentation, and asked if you could comment a bit more about changes, prenatal changes for the female brain.
Oh, no idea, because I don't even know whether those donors, for example, for the single cell RNA, these were 5 donors, right? So you can't make much statements about sex differences there. For the brain span, I think that from what we have seen when we were doing virtual histology, the sex is split more evenly, more or less half and half. But again, the numbers are too small to get at the more subtle differences that would exist between sexes. I bet you that the differences between regions are of the order of magnitude or more than 1, larger than what we would see as differences between the 2 sexes. So I think that's another call for more data to be able to then start reanalyzing everything along those lines. Now, what we can do, and we have not done, you saw that in both Enigma examples, we use sex as a confounder. Now, we could have, of course, we could have split those large samples based on sex. But remember, this is, for example, for the virtual histology, it's 140 cohorts. So that's a lot of technical variation in terms of the scanners, recruitment, diagnostics, etc. So to do it properly, if we wanted to compare males and females, we would have to make sure that we have the same number, or more or less, same number of males and females from each acquisition site, because we don't want to be comparing males from one side with females from another acquisition site. So it gets challenging, even with those large numbers.
Great.
Oops, sorry.
He liked your presentation. So Myrna Weissman wrote in, the effects of the environment are difficult to test with these methods, but have you looked at the effects of toxins, for example, SSRI exposure during pregnancy?
No, we haven't done any work along those lines. I mean, in the SARGON-AU study, we designed a study to look at associations between prenatal exposure to maternal cigarette smoking and adolescent brain and body. And we reported quite a few associations along those lines, but we never pursued in this particular cohort a relationship of the sort that Myrna mentions. Remember, these are community-based samples, right? So we are not recruiting from populations that would have particularly high rate of, for example, exposure to SSRIs or other medications. Now, of course, what we are going to do and others are doing, once you have those large community-based samples, you can start looking at the relationship with physical or social environment at an aggregate level. So ABCD study is doing, for example, work with air pollution, right? So you can, of course, use government databases to estimate the levels of air pollution in the areas in which your participants come from, and then link, compare participants as a function of the aggregate level geospatial unit coded air pollution. And I think that kind of work is going to be very helpful in the future, that you can mix the individual level phenotyping with aggregate level exposures.
Yeah, that type of geocoding is also being done in the ECHO study, which focuses on a younger population, but I fully agree, it's very, very fruitful. So I think that probably will have to be our last question as we're approaching 12:30, but I wanted to give Dr. Lieberman a chance to have the final words.
I'll sneak in a question with that. So the word, you know, the source of your gene expression data is postmortem tissue. Have you tried or thought about using iPSCs or organoids to be able to have more variability in terms of the kinds of effects you could look at?
Yeah, we have haven't done so. Have tried writing some grant applications to get money to do exactly that, but on a large scale. In epidemiological studies, it's still not feasible, right? I mean, ideally, it would be great if we could get iPSCs from tissues, fibroblasts, or blood cells for each of the participants. But I think it's too expensive and too premature to do it this way. So I think we are far away from doing it in this context. I think iPSCs are working much better when you have a very clear, far apart case controls than in the context of course, psychiatric disorders. People have done it with schizophrenia, bipolar, etc., but the samples are typically relatively small. But it's certainly an avenue to pursue in the future.
Well, Tomas, thank you for a wonderful presentation and very, very just exquisite work. And we look forward to following up with you, you know, quickly and regularly. So thank you.
Thank you.
All right, Jonathan, good job. See you next week.